Okay, that was a fantastic session. And questions? Yes, go ahead, Ross.
This is a selfish question. Are you going to put those mice on CR and look at the mitophagy?
The chemo mice.
Yeah.
I'll just repeat the question. Is there any chance you'll put those mice on CR and look at rates of autophagy in different tissues? My suspicion is that there's a discoordination in rate of aging in the rapamycin model that you may not see in a caloric restriction model.
Yeah. So we did the opposite experiment. We fat-fed the mice, and we saw that that produced a decline in mitophagy, but we've not done the caloric restriction. It's a good experiment. We did cross the mTOR hypomorphic mice with the chemo mouse. We were hoping to see higher rates of mitophagy, and we couldn't convince ourselves we could. So I don't know what that means. I don't know what the sensitivity of the chemo, I don't know how much we can, I don't know if it's a sensitivity issue or whether or not, you know, I don't know. We don't. Yeah. So
the other possibility is if the pathway is really important developmentally, which it may be, because now there is this suggestion that autophagy, and that's something that we're looking at, that autophagy actually has a crosstalk with synaptic vesicle homeostasis, then, you know, it could be that you've, you know, downregulated or, you know, neutralized that pathway developmentally. So did you give rapamycin to these animals?
No.
Okay.
No. Everybody's tired.
More questions? Okay, then I'll ask one question to Toren. Is there any evidence, apart from the rapamycin studies, that mTOR1 and mTOR2 have a differential effect on rate of protein biosynthesis and rate of autophagy?
Wait, so the differential role of mTOR1 and mTOR2 in
regulating rate of protein biosynthesis and regulating autophagy.
Yeah, I mean, mTOR1 is generally thought to be regulating protein synthesis. mTOR2 biology is a little less understood, but its outputs are generally thought to be cytoskeletal and other things. So mTOR1 is what rapamycin generally, I mean, it's complicated, but there are feedbacks between mTOR1 and mTOR2, but the general target of rapamycin is thought to be mTOR1. And
so is that evidence just coming from the rapamycin studies, or there is any genetic proof?
There's David Sabatini's group has done some genetic proof of the role of rapamycin and the beneficial versus harmful effects in terms of glucose tolerance being mediated by different mTOR complexes.
Yes, go ahead. Ross, again.
It's her again.
So I'd like to ask this across the panel. So one of the things we see when we look at aged tissue is a huge increase in variance in parameters. And it's bad enough in hybrid mice, but it's appalling in monkeys. But I think that there are possibly biological insights to be gleaned from that. So do any of you incorporate this type of thing into the way you analyze the data, particularly in the old models?
Actually, we've noticed, and this is anecdotal because we haven't really done anything formal, but we've noticed throughout in most of our measures that that's one of the things that, you know, called my attention, let's say, looking at data, first, you know, large behavioral cohorts of all sorts of measures of cognitive and non-cognitive, et cetera, but it even goes down to biochemical data, protein abundance, that the variation seems to be collapsed in animals that have been treated with rapamycin. And this is like the natural aging variation. Not the Alzheimer's disease is a different, you know, those models are different. But in the studies, the few studies that we have done, a variety of measures when we look at the non-transgenic animals, for instance, and we have like, you know, advanced ages more than, you know, 12 months or so. But when it really gets to high, you know, much older animals, we see a reduction in that variability, which seems to be fascinating, but, you know, it's hard to put a finger on as to how that comes about.
I suspect
go ahead,
that
go ahead, please.
as you approach the extreme of just about everything you're going to look at, you'll see an approach to less variability. So that with the supercentenarians, they're an incredible, aging itself is a very, as you say, extremely heterogeneous. And all you have to do is, maybe not in this group, but in other groups, look around you and you can see how differently people age. But when you get to these most extremes, like supercentenarians, they become very alike. These are the folks who are markedly delaying their disability and morbidity towards the very end of their lives. And you don't see much variation in that, whereas you do with the 100-year-olds, people living to 100, 101. And I think that's, it's almost like a mathematical property that as you get to the most extreme of a Poisson kind of distribution, the things at the tail on either end are going to be much more alike. And I think we see that with the supers, which speaks to the fact that they probably have some really interesting genetics in common with one another, or other biological mechanisms.
Maybe I could ask a very naive question, but I think many of us are interested in the crowd. So when can we start using rapamycin to extend human life?
That will be Torrance.
Well, all I can say is that in mice, it produces testicular atrophy. So I think half the people are not interested already for doing it. I mean,
Trade-off.
There are, I mean, like I said before, at least in our genetic model, there are trade-offs, in terms of you live longer, but, we saw more cataracts in the hypermorphic mice, bone density went down faster. So, I mean, I don't think it's the perfect drug.
But do you have to worry about testicular atrophy when you're 105?
Well, it depends.
I'll take it.
On the same line, many of us are aware of interest in metformin because you're talking about longevity, and Neil Barzilai's study is well known and is about to be launched as a human trial, both for MCI prevention as well as longevity of human life. Could you please care to make a comment on metformin and longevity for me?
I think the epidemiological evidence is very interesting. I mean, I think, as probably a lot of people in the audience know, that, just looking at diabetics who take metformin versus diabetics who don't take metformin, the diabetics who take metformin appear to live longer. And actually, if you look at people who are diabetics who take metformin versus people not taking metformin, there's evidence that they live longer. So that's pretty, I mean, that's not a prospective study, but I think that's very interesting. Clearly, Veronica's, AMP kinase ties into the mTOR pathway, and energy sensing is very appealing. I mean, I should say, metformin is thought to work via AMP kinase, and it's thought to produce a sort of complex 1 inhibition and produce sort of a mitochondrial dysfunction, which may trigger some protective sort of hormetic effects in tissues and organisms. So, I mean, I think the mechanisms are enticing in sort of a cell biology perspective. I think the epidemiological evidence is also very intriguing. I don't do clinical trials. I don't know if this is the best way. The design of the trial is necessarily the best way. It's certainly expensive, but I think something like that makes a lot of sense to begin to try. I think interrogating aging or aging-related pathways with small molecules, I think is where aging has to go, because I think we've built an incredible base of knowledge but a lot of the knowledge has been sort of correlative, and the question is, can you take those insights and make predictions and design molecules that affect the relevant pathways, and can you have an effect? And whether that effect is going to be on lifespan or health span or stress resistance, or I don't know what the best design is, but I think it's good to get the field to a point where we're moving beyond, sort of correlating what goes and trying to make using that knowledge to sort of make rational interventions. And I think metformin fits that category.
Anita.
So I have a question about a different kind of, about Subhajit's idea of how to stave off Alzheimer's disease and maybe increase longevity. So you did a really nice job of describing how specific the CRISPR-Cas9 system can be. But can you speculate about, or have you thought about really how specific it is? How would you be confident that you're not getting any off-target effects? And, or do you think that that will come as the technology gets better, or it's still a young technology? Because I think that the idea behind it, behind being able to just clip off that little piece of APP and you can really change the way the cells handle it, the APP is really, really provocative.
Yeah, so I mean, I actually don't know, because what you don't see, right? So I think there may be some off-target effect, but you probably know this better than me, but you can do all these bioinformatics to predict off-target effects. The one thing I was really worried about is that APP, for example, has these two other genes that compensate for it, which is APLP1 and APLP2. And APP knockout mice are actually kind of fine, more or less, but if you knock out two of the others, then actually the mice dies. But it turns out that, at least from the bioinformatics that we've done, it seems that our CRISPRs should not have an effect on those other two based on just the sequence. But I think this is, from what I understand, even the CRISPR experts struggle with this. What do you really do? I mean, you basically could do a whole genome sequencing of these cells to find out every single gene that has been modified, but you can't really do that. So the approach that we are taking is that we will do as many physiologic tests as possible. But the other thing to think about is that you can't have the perfect drug, right? I mean, you have this testicular atrophy if you take, whatever. So I mean, if you really have Alzheimer's disease and you have a brain that's going to be, chock full of plaques in 5 years, would you take this CRISPR and block the further progression of amyloid? I absolutely 100% would do it, if I was convinced that at least whatever I have seen, there's no effect on axon growth, dendritic maturation, and in vivo testing, which we haven't done yet. We are kind of in the process of doing it. So if all those pan out, I think I would take the risk. I don't know. But
Ken.
I don't know if Suchan knows more about it than
I had a question for Tom. I was wondering if you could expand a bit more on your observation that many of the centenarians, if I'm getting this right, are getting the diseases but not getting the disabilities that go along with the diseases. And it's sort of analogous to the cognitive reserve idea that folks are getting the Alzheimer's pathology, but somehow they're able to compensate better. I was wondering if you could just expand on what are the pathways or what you're thinking about.
It's a wide open field, and it speaks to functional reserve and adaptive capacity. And I think there's very little known about the anatomic and physiologic correlates of those things. So I think that field is wide open for people to study, and I think it's a very hot topic in gerontology.
Yes, Andy.
Yeah, follow up to Ken's question. Have you looked at the brains of these supercentenarians?
Yeah.
The problem is, Claudia K. Watson in the 90s, the pathology has no prediction on metabolic performance at all. In that group, it's just the pathology is all over the place.
You bring up a really sore subject because you don't know what you got. I work with a guy named Lee Goldstein, who is an ophthalmology kind of guy. He's a Beeson person, and we told him one day that there's a portion of our centenarians who never got cataracts, and they still have their natural lenses. And a 106-year-old lady just passed away, and he didn't believe us, and we got her eyes at autopsy, and she had her natural lenses with luminescence or kind of the clarity of the lenses of a 50-year-old, and it was really interesting. And it would have cost me $1,500 to also get her brain. And it was also a problem that we didn't have cognitive testing on her before she died, because the family had just come to us asking, they said that their mother was passing away, and they thought it would be a tremendous waste to not donate her body to science. And we get requests like that, but it's very expensive. And so we made the decision not to get the brain, and I'm really, really sorry that we did. I think, anyway, so we have 56 brains banked. But unfortunately, one of the problems with being at Boston University is it's also the biggest center for traumatic brain injury and working with NFL players. So there's no resources to work on our brains. So we need to get a grant that will pay for a neuropathologist to work on those. When we were at Mass General Hospital with a brain bank there with Tessa Headley-White, we did a small series of 14 brains, and about 15% had no neurofibrillary tangles or plaques, and BRAC scores of 0, or basically CDRs of 0. And then there were people who had Alzheimer's and had brain pathology consistent with that. And then there was a group in between that had lots of neurofibrillary tangles and plaques that should have been like a BRAC that was way up there, or a CDR of 1 or 2, but they had CDRs of 0.5. So there was this discordance between the pathology and their cognitive function that goes along with this cognitive reserve or adaptive capacity.
Are you looking at the lens with Lee?
Sorry?
Is Lee looking at the lens?
Yeah, so there's this really interesting thing. I won't take up too much of your time because I also have a flight to catch, but the lens of the eye has clear cells in it that are kind of like the layers of an onion. Those develop at the 5th month of fetal development, and they don't change over the whole lifetime of the human being, except there's some protein changes. And it's thought that those protein changes that you can assess through light spectroscopy, and the way that the light bounces off of these proteins can be a biomarker of aging. And so, depending upon that, those accumulated protein changes, the hypothesis is that those can be very different in these centenarians. And what he needs, though, is the natural lenses. And the problem is that many older people get cataracts, and they then get these artificial lenses, and there goes you don't have biomarkers anymore. So we get him subjects, hopefully, that still have their natural lenses, and he can assess that.
Yes.
I know someone has a flight to catch, but I just had a question as a clinician in the room more so than a researcher. I'm just curious, we've certainly heard mention about the calorie restriction, and I know you talked about in your study participants, you've got the men who are typically, trim and thin, whereas the women perhaps are shorter and stockier. I'm just curious, as our whole patient population is getting more savvy with kind of reading research and surfing the internet, just kind of, do we have any thoughts about, okay, calorie restriction and in what ways? And why is it that the men do better when they're trim and thin, and the women seem to do better, perhaps being a little overweight? Is there any wisdom you'd share about what you'd talk to patients about when they come to you with that?
No, I would just say to a number of people in this room that I think they need to study men versus women at these ages and see how their pathways around caloric restriction, mTOR, and sirtuins are different, because I think they're going to be. And I think it's going to be terrifically interesting.
Okay. Oh.
Just wanted to echo, I think many people here have said that the clinical expression of dementia is very complex, and it's not just a function of the pathology which is present in the brain. And I think in the morning session we heard about the complex interplay between the social, the psychological, the socioeconomic, the epigenetic, and many other factors which interact with the pathology in the brain and then eventually result in the expression of the disease. So I think we have to really understand how other factors that affect clinical expression, what pathways do they affect? And unless we understand that, I think the eventual clinical phenotype of the disease and its prevention may not be that possible. We can target the pathology, but there are many other factors that play into the eventual expression, and I think a lot more work needs to be done that Ken just pointed out.
So one point about pathology is that you see what you know, right? I mean, we look at plaques and tangles, but we may be missing, tons of other protein pathology. We don't really check for them. So I think if you have an abnormality, I would think that you would have a pathology associated with it, but I would maybe think that our tools are not good enough. I mean, it's not like sequencing where you can just put the brain and know every single pathology in the brain, no? So maybe that's one comment.
One more question, yes.
In terms of the delta-delta M2R mice,
Yeah.
So there's a significant shift in the transcriptome of CR mice versus control mice. Is there?
We didn't do any transcriptional analysis, so I can't say. Other people have, but not in this model, but others, but we can't say what the overlap is.
Okay, I think we can stop here. Thank you again for this very nice session.